Welcome back to BAP e-Learning Center. In our previous lesson, we explored the glossary of genetics. Today we advance that journey as we dive into monohybrid inheritance, one of the foundational pillars of Mendelian genetics. My name is B. Brian Bahundira. Remember to subscribe, like, and share so you never miss any of our enlightening science lessons. Today we are looking at the monohybrid inheritance. This is the genetic study that involves inheritance of only one trait controlled by a single gene with two alleles. In our previous video, we defined allele as the alternative form of a gene, and monohybrid inheritance can better be explained by the Mendel's first law of inheritance, or at times it is referred to as the law of segregation. This word segregation is derived from meiosis, whereby the homologous chromosomes in meiosis 1 separate during anaphase stage of meiosis 1, resulting into formation of chromosome, each chromosome carrying two alleles occurring on different sister chromatids, which also later separate, resulting to formation of gametes, each gamete carrying a single chromatid, each chromatid having a single allele. Let us look at Mendel's first law of inheritance. It states that the characteristics of a diploid organism. A diploid organism, this is an organism with two sets of chromosomes, 2n, where n is equal to 23 chromosomes and 2n resulting to formation of 46 chromosomes. A diploid organism are controlled by alleles which occur in pairs in the body cells but singly in gametes. Each offspring then receives its own pair of alleles for that trait, one from either of the parent. Whenever of the two alleles in the offspring is dominant, it determines the phenotype of an offspring. How meiosis explains Mendel's first law of inheritance. The genes determine organisms' characters usually occur in two alternative forms. Those are alleles. The two alternative forms are the alleles, called alleles, located on homologous chromosome. During anaphase 1 of meiosis, these homologous chromosomes separate and move to the opposite daughter nuclei. Subsequent cell division, that is meiosis 2, results in two gamete cells, each containing one of the two alleles. Therefore, the alleles occur as pairs in body cells but singly in gamete cells. This is an illustration that indicates how segregation occurs. These are the homologous chromosomes during meiosis 1. Meiosis 1 results into formation of chromosomes, each chromosome with two sister chromatids, each chromatid carrying an allele. During meiosis 2, the sister chromatids separate or segregate, resulting into formation of gametes, each gamete containing a single chromatid, and each chromatid carrying a single allele. This is Mendel's first law of inheritance explained by meiosis. Let us look at the sample question that show the derivative of the Mendel's genotypic ratio and the phenotypic ratio of 3:1. In plants, tall T is dominant over short. A homozygous tall is crossed with homozygous short. Question 1. Show the genotypes of the parents' generation. P stands for parental generation. Question 2. Work out the genotype and phenotype of the F1 generation. B. The F1 plants were selved. Work out the genotypic and the phenotypic ratios of the F2 generation. Let us start with question A. Our question A reads, a homozygous tall. Homozygous, it simply means that the alleles were similar. Tall is crossed with homozygous short. That is A, our first question. Show genotypes of the parents. Let T represent allele. Allele for shortness. For tallness. For tallness. And let T represent allele for shortness. That is solution to A, Roman numeral 1. So Roman numeral 1 was asking for parental phenotype. Show the genotypes of the parents for parental genotype. Let us first look at the parental phenotype. Parental phenotype. This is tall was crossed with short. The parental genotype is homozygous. Homozygous tall crossed with homozygous short. So when we write it in with alleles, we shall have the parental genotype, will be homozygous tall and homozygous short. These are the parental genotypes. We look at our Roman numeral 2. Work out the genotype and phenotype of the F1 generation. Obtaining the F1 generation, we are going to cross the parents. There will be fertilization, whereby a homozygous parent for tallness met with a homozygous short parent. Let us consider a tall parent to be a male and a short parent to be a female. This is Roman numeral 2 of A. Parental genotype. We have homozygous tall crossed with homozygous short. Here the alleles exist in pair, meaning that these are not yet gametes. They are still body cells. Then Mendel's first law of inheritance, it explains the formation of gametes. So here we are going to form the gametes. Gametes are formed by the process known as gametogenesis. Whereby the alleles, a pair of alleles, separate into single alleles in gamete ready to undergo fertilization. The homozygous short also undergo meiosis, forming gametes which gametes later undergo fertilization. Fertilization, one gamete fertilizes the other. This is the male gamete, these are the male gametes, and these are the female gametes. The male gametes are fertilizing the female gametes. This is a sperm, it fertilizes an ovum, forming a zygote. And this sperm also fertilizes this, forming a zygote here, or an offspring. This one also does the same. It also reacts with this. So here we obtain the F1 generation. This is the F1 genotype. And I hope our question was asking the F1 genotype. In case you were asked also to set the F1 phenotype, so the F1 phenotype would be all offsprings are tall. Show the genotypes of the parents of the parental generation. Then we're from solving Roman numeral 2, which was work out the genotype and the phenotype. Fine. The phenotype was also asked. So we have solved Roman numeral 2. We are deriving the monohybrid ratio, the Mendelian ratio of monohybrid inheritance. Let us go through B. The F1 plants were self-pollinated. Work out the genotypic and the phenotypic ratios of the F2 generation. These are the F1 offsprings. These are the offsprings and B, it's saying these offsprings were self-pollinated, meaning that a sister met with a brother. So let us have B here. B. Now they are becoming the parents now. Parental genotype. They are now heterozygous. Heterozygous tall man crossed with a heterozygous tall woman. We shall have T, the genotype, crossed with T. We come to the next step is formation of gametes. We apply Mendel's first law of inheritance or the law of segregation. Here gametes are formed. These two separate, forming T and T. These two also separate. They undergo segregation such that they form gametes. Each with a single allele, such that during fertilization, a diploid condition is retained. Fertilization. Now this is the F2 generation. Our part A was the F1 generation. This one fertilizes this, forming homozygous T. It still fertilizes this one, forming a heterozygous tall. This is homozygous tall. This is heterozygous tall. Why? Because all the genes here are dominant, so producing a tall offspring. Also, here, the allele for tallness is dominant over the allele for shortness, which is recessive. Thus, it masks the effect of this allele for shortness, resulting to producing of a tall offspring. This recessive allele for shortness fertilizes the allele for tallness, forming a tall offspring. It also fertilizes this one, a recessive allele fertilizes another recessive gamete, resulting in formation of a short offspring. This is the F1 genotype. F2, I mean to say, this is the F2 genotype. So the question was asking for the genotypic and the phenotypic ratios. How do we get the genotypic ratio? The phenotypic ratio is got from these alleles, from the offsprings. We see that the offsprings will have tall, 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 and then short. The genotypic ratio is 1 T one TT 2 T then 1 T. Then phenotype, the phenotypic ratio. We shall have this one is tall because it contains two dominant alleles for tallness. This one is tall. These ones are also tall because they contain a dominant allele for tallness, which masks the effect of the recessive alleles. Thus, the offsprings become tall. However, this one, it contains two recessive alleles, and they all express their effect, resulting into an offspring being short. Thus, we have the phenotypic ratio is 3 tall because this one and this are tall, then 1 short. This results into a monohybrid phenotypic ratio of 3 to 1. Thus, we are done with our question. Work out the genotypic and the phenotypic ratios of the F2 generation. Thus, the genotypic ratio is 1 TT 2 offspring. They have a dominant allele for tallness and a recessive allele for shortness. then one offspring, one has double recessive alleles for shortness. That is the genotypic ratio. Now, the phenotypic ratio, 3 offsprings are tall and 1 is short, which gives 3 to 1. You can pause and understand well where you have not understood, or if you are noting, you can pause and note. Here you can use here. Now you can test your understanding. You can put the answers in the comment section, or we have a WhatsApp channel where you can put your answers at 0770 97 69 74, or you can email us at bablearningcenter@gmail.com. In this question, there is the use of a Punnett square, which is also okay. A Punnett square, it means here epiplant, tall, thin, and that. So it means that after the formation of gametes, the mating takes place using a Punnett square. Takes place using a Punnett square, whereby here the gametes will be in piplant, tall is dominant over short, a homozygous tall, okay, homozygous tall, and heterozygous tall, meaning that a parent was dominantly tall and this one was heterozygously tall. So the gametes will be tall, tall, this one tall, this one short. So these gametes are the ones that are put here in this square. This is our first gamete, this is the second gamete, and then this one is put here. These ones are these. These ones, sorry for that, these ones are these, and these are here. So here you will fertilize like this. That is a Punnett square, and it looks more simpler than the crosses. That is. Let us come to the modifications of monohybrid inheritance. These refer to situations where Mendel's expected phenotypic ratio of 3 to 1 in the F2 generation does not appear because the genes being studied do not follow the simple complete dominancy. The first modification, we have codominancy. The modifications here, they are conditions that alters. These are conditions that alter the Mendel's phenotypic ratio of 3 to 1. You may find that instead of forming 3 to 1, there is a change in the phenotypic classes. You find that the phenotypic classes are 3. Here the phenotypic classes are 2. But there are conditions where we find that the phenotypic classes are becoming 3, like 1 to 2 to 1. Shall see. Let's continue. Codominance is our first condition. This is a genetic phenomenon where a cross between pure breeding, in the glossary we also looked at this term, pure breeding. This is where organisms that consistently show the same characteristic from generation to generation are made. That is pure breeding. This is a genetic phenomenon where a cross between pure breeding parents with contrasting expressions or different phenotypes for the same character. This statement means parents with contrasting expressions for the same character. The character might be tallness, that is, the character might be height. Then height is divided into being tall and short. That is pure breeding parents with contrasting expressions. These are the contrasting expressions. One can be tall and another can be short, but of the same character. Here the character is height. Or contrasting characteristics, it can be skin color. A skin color, one can be brown, or can be black skinned. This is the same character but with alternate expressions or with different expressions. For the same character, produce an F1 progeny with a phenotype which is a mixture of the two parents. Both alleles are expressed equally in the phenotype of the offspring. They show a mixture. For example, if a mother is brown, then a dad is black. Then they produce a progeny or an offspring, or they produce a daughter or a son with both black and brown patches. However, this one does not happen in human patches. It does not happen in humans. However, it can happen in animals like cow, goats. This condition changes the phenotypic classes from 2, that is 3 to 1, to 3, 1 to 1 to 2 to 1, like I earlier explained. Examples of genes which show dominance include inheritance of coat color in cattle. We shall look at an example. Genes for the A and B antigens in man. These are blood groups. They can show codominance. For example, I think this example may not look at it. However, I shall look at this and I can bring it here. For example, A this is an allele for A, blood group A, crossed with that for blood group B, heterozygous blood group A and heterozygous blood group B. Here you can get offspring with blood group. This one meets with this or fertilizes this, forming AB, blood group A, B. This one still with this, forming A, then this one with this, forming B, or then this one mixed with this, forming, find that here we are having 3 offsprings now. Offspring with blood group AB. This is blood group A, and this is blood group B, and this is blood group O. You find that all are present here. The phenotypic ratio has been altered. The other example is genes for hemoglobin synthesis in man. Hemoglobin synthesis in man. We shall look at it, how it does that. Note, alleles showing codominance are represented by different uppercase letters as illustrated in the subsequent pages. Sample question that show how codominance alters or changes the normal Mendelian phenotypic ratio of 3:1. In cattle, coat color shows codominance. Alleles R is equal to red and W is equal to white. Heterozygote WR is equal to Rowan. Rowan is a mixture of white and red hairs. A pure breeding red bull is mated to pure breeding white cow. State the genotype and the phenotype of all the F1 offsprings. Show the cross used to get the above answer. 2 F1 Rowan individuals are mated. Show the cross using the cross probabilities. Calculate the genotypic and the phenotypic ratios of the F2 generations. We'll come to the question interpretation. Here, a red bull. This is a red bull as indicated in our question. Red bull. A red bull met with a white cow. It was represented by W. White cow. It gave a rowan calf, a calf with both red plus white patches. This is a calf, a younger one of a cow. A red bull, when crossed with a white cow, it gives a rowan calf with both red and white patches. Our first question requires state the genotype and the phenotype of all the F1 offspring. A. Let R represent allele for red coat, coat color, and W represent allele for white. The question requires the genotypic ratio, the genotype of the F1 offspring. The genotype would be R double U, and the offsprings are 4. That is the answer. Then our part B was asking for it is part Roman numeral 2 of part A. It was asking for the crosses. This is the parental, genotype. Whereby you have a red bull crossed with a white cow. We come to formation of gametes. We have R and R crossed with white here. These are from the segregation. Then we go to fertilization. Fertilization occurs whereby R fertilizes W, forming R, forming an offspring, producing offspring, which is a raw one. This R also fertilizes a white cow, forming a raw one offspring with both red and white punches. Still, this red bull fertilizes, forming R, a calf with white and red punches. And the F1 generation results into formation of 4 offsprings, each with red and white punches. This is the F1 genotype. I think this is what we have here. That is. When we cross, I think we are looking for how codominancy alters the 3:1 monohybrid genotypic ratio. In looking for this ratio of 1 to 2 to 1, we are now going to self these offsprings. Let us consider these ones. F2 generation. The offsprings now here, they become the parents. Those are parental genotype. We have R, a raw one father crossed with a raw one mother. We form gametes. Gametogenesis occurs another gamete, one of the gametes from the father will be containing a red, an allele with a red color, and the other will be having allele controlling the white color. Then also from the mother, one of the alleles one of the gametes contain allele that controls the red color, and the other gamete contains an allele which controls the white color. Fertilization occurs we form, this gamete fertilizes this, resulting to formation of a homozygous offspring with red alleles. This very gamete fertilizes this one, resulting into formation of an offspring with both red and white color controlling alleles. This one fertilizes this, resulting formation of an offspring with both red and white color controlling alleles, and this gamete containing a white color controlling allele fertilizes another gamete containing a white color controlling allele from the mother, resulting in formation of an offspring with two alleles which control the same color of white. That is the genotype F2 genotype, the genotype that results from selfing the offsprings. Genotype. This is the genotype. What is the genotypic ratio? Genotypic ratio, we have 1 R 2 R W and 1 W. Thus, the phenotypic ratio, is 1 red, 2 Rowan, and 1 white. This condition results into changing of the phenotypic classes from 2 to 3. These are 3 phenotypic classes, 1, 2, 3, and these are 2 phenotypic classes, 1, 2. That is how codominance alters the normal genotypic ratio in monohybrid inheritance.